So,
Yes, sir.
any questions you people want to ask?
सर क्वेश्चंस पे
मामा से एनसीईआरटी लेके आया।
तो आप क्वेश्चंस पूछोगे जैसे कि हम बताएंगे।
Online batch, you people have any doubt to ask? Gayatri, Devrat? Nothing, no questions. आपके नहीं आते बच्चे का? मन है वो, मन बड़ा जालिम। Yeah, it's taking a while to open the PDF and then we'll start. Today we are going to see the genetics chapter from NCERT point of view, okay? Earlier also we are doing the same thing, but now we are going to see the directly NCERT. Okay, so before we start, I like to share that whenever you are doing NCERT, it means you have to do this part also. So for every unit they have said something, right? You don't have to ignore this part, because if you see they have explained the factors here only, right? So for every unit they have given some information here, so that what in that particular unit you people are going to do, right? Please do that part also. Going to next topic. Here for every this they'll give this also for every here they have given the Watson and Crick because they have received Nobel Prize in there, right? What they have done?
Double helix model.
Double helical model they have proposed, right? So please read that also. Important. Coming to the chapter 5, and how many people have scanned this one and seen what is there? Anyone?
वैसे ही कुछ इन्फॉर्मेशन होती है, एक दो पेजेस ज़्यादा होती है पिक्चर्स और उसमें से
Exactly. So
डायग्राम.
please scan this particular part and see what they have given. Okay, so sometimes they may frame a question from there also. Basically they have included practicals, right? So please scan and see what they have said. Okay, so shall we start?
Yes, sir.
मन तो आएगी, मन तो
नहीं करिए ना इस सर मैं वहाँ बैठ जाऊँ.
अच्छा हो. So starting with the introduction, what they said is, have you ever wondered why an elephant always gives birth to only a baby elephant and not to anyone else? The mango tree is always going to produce a mango tree. Why? This is class 10th. What could be the answer?
Because the traits are passed down by जो traits
उसके जो
हैं
जीन्स पास ऑफ हो गए वही तो बनाएंगे.
हाँ
जीनोम होता है ना.
elephant के genes
Okay.
pass down हुआ तो elephant ही बनेगा.
Inheritance, because of inheritance, right? What people are studying in a sentence. Right, so because of inheritance, this is there. Given that they do the offspring identical to their parents, or do they show a difference in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These and several related questions are dealt with scientific in this branch of biology called genetics, blah blah blah. Now this is important. This subject deals with the inheritance as well as variation of character from parent to offspring. Right? So inheritance means what? Inheritance means what? What is passing from parent to offspring is called inheritance. Now variation means what? The degree of change. Degree how much the children is differ from their parent. That's called variation. And how a variation is going to be arise?
recombination.
One is recombination and second is exactly. Then inheritance is the process by which characters are passed on from parent to progeny. It is basis of inheritance. See this, how many times they have said the same thing. Basis of inheritance for this also, for gene also, for chromosome also, right? Unit of inheritance, basis of inheritance. Variation is the degree by which progeny differ from their parent. Right? Same sentence in class 10th also there. Humans know from as early as 8,000 to this much that one of the cause of variation was hidden in the sexual reproduction. And what is the cause? What they are discussing here? Crossing over, right? The meiosis part, not the mutation part they are discussing. Yes or no?
Yes, sir.
At which stage we have crossing over? Good. Packing. We have to be specific, right? During packing of prophase. Then nothing is there. Coming to this part, Mendel law of inheritance. Now few points here are important here. It was during the mid-19th century that headway was made in understanding inheritance. George Mendel conducted hybridization experiment on garden peas for 7 years. Now try and understand. How many characters he has taken?
7.
7. And for how many years he has done the experiment?
7.
Please remember that. And its nature of experiment is what? Hybridization experiment. What he has done? Hybridization experiment. Please remember. And he proposed law of inheritance in living organism. During Mendel investigation into inheritance pattern, it was the first time that statistical analysis, mathematical logics were applied to the problem in biology, and that is the reason why the contemporary parts didn't accept his idea of what he has find out. His experiment had a large sampling size. Now, why it's true? Because of large sample size. Yes or no? If you are proving in a single individual that this is happening, people won't believe. If you prove in thousands, people will believe that it's not in one case, it is happening because of some reason. So he has taken a large sample size which gave greater credibility to the data that he collected. Please remember this. From also the confirmation of his inference from experiment on successive generations of his test plants proved that his results pointed to general rule of inheritance rather than being unsubstantial idea. What is unsubstantial?
बस मन से बना दिया कि ऐसा
Without any proof. So whenever he has performed the experiment, he got the same ratios which give a particular thing that whatever he is saying is true to the very fact and it's not making up the facts. Right? So 2 points to his success. What is one? The large sample size gave a credibility, and every time he had performed the experiments, he got the same results. Right? That's what. Then Mendel investigated characters in the garden pea plant that were manifested as two opposite traits. One is tall and second is dwarf, yellow or green. So what they are saying that whatever the characters, 7 pairs of character he has taken, it means they are contrasting to each other. Clear? Then this allow him to set up a basic framework of rules governing inheritance, which was explained on by later scientists to account for all the diverse natural observation and complexity in inheritance. So the people who worked after and still are using the same idea that a gene is there, it can be either going to be expressed itself or not going to be expressed itself, right? Dominant or recessive. The same very thing are people using again and again. Yes or no? The Morgan, same thing, right? He able to the chromosomal theory of inheritance, same thing, right? So people have done this over and again. Because of that only Mendel is called as father of. Mendel conducted, yes sir? Mendel conducted such artificial pollination, cross-pollination experiment using several true breeding plant line. He used always true breeding. True breeding means what? On and touch, people are getting
homozygous gene.
Manal, Gayatri, Devrat, Bhavika, Anjana. Sleeping, eating, biting. No response. Online.
No, sir.
Achha.
No, sir.
Okay. Hey, you're not getting. Gayatri, you're not getting there.
आप लोगों का खाना है तो
Oh, for other. Okay, good. A true continuous, a true breeding line is one that have undergone continuous self-pollination. This you have to do in when you are doing with the economical botany and economical geology. Right. Show that stable traits, inheritance and expression for several generations. Mendel selected 14. How many?
14.
14 true breeding pea plants. So he's taken 7 characters, done the experiment for 7 years, and taken how many plants?
14.
Only 14 plants or 14?
True breeding people.
Right. So the different type of plants they has taken is 14. Why? Each one. 1, 2, 3, 4, 5, 6, 7, 8. Set. Right? That's why. But a large, thousands and thousands. Right? So I think he has counted 16,000 some seeds to get to a conclusion of 9:3:1 ratio. Right? See how much free time they have. As pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkle seeds. Blah 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 blah. Done. Already asked, still important. Done. Which is there for contrast, dominant or recessive. Now what he has done, a process called emasculation, where you have to remove the ovaries, right? Anthers. Removal of anther is called emasculation. And covering of
Bagging.
exactly. So that's how they pollinated and done with the result. Now inheritance of one gene. What you have to study here? Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study the inheritance of one gene. He collected the seed produced as a result of this cross and grow them to generate plant of first hybrid generation. This generation is also called filial generation. And I told you people, filial means what? Offspring. Filial means what? Offspring or progeny. The Mendel observed that all the F1 progeny plant were tall. And I told you from here what he got? F1 law of dominance. That the dominant character is going to express itself in F1 generation. Yes? Like one of its parents, none were dwarf. He made similar observation. Again, I thought somewhere is talking from there. He made similar observation for other pair of trait. He found that F1 always resembles either one of the parent, and that the trait of other parent were not seen in them. Mendel then self-pollinated the tall F1 plants, and to his surprise found that in F2 generation some of the offsprings were dwarf. The characters that were not seen in the F1 generation was now expressed. How come? Because from this experiment they have nullified the rule of blending. That nothing is blended. The things are going to be present at discrete unit and they are going to segregate again. Okay. The proportion of plant that were dwarf were 1/4 of the F2 and 3/4 of the plant at tall. 3 more. The tall and dwarf traits were identical to their parental type and did not show any blend. So whatever he got in F2 generation, no blending was. That is, all the offspring were either tall or dwarf, none were of in between height. Time increasing from Serene. You are there, dear? Siri. Thank you. Okay. Similar results were obtained with other traits that he studied. Only one of the parental trait was expressed in F1 generation, while at F2, both traits were expressed in the proportion 3:1. The contrasting trait did not show any blending at either F1 or F2. What is clear? You people online batch getting? Online batch?
Yes, sir.
Okay, dear. Now, based on these observations, Mendel proposed that something was being stably passed down. Stable passed down. Means not changing from generation to generation. Yes or no? Not changing, not blending, and getting expressed. From parent to offspring through the gametes. Right? Yes, yeah. Gametes are only fusing. Over successive generation. He called these things as factors, now we call them genes. Genes therefore are the unit of inheritance here. Basic unit of inheritance, now unit of inheritance. Okay, and then chromosome also you will get somewhere. They contain the information that is required to express a particular trait in an organism. Okay, it means what? They are getting expressed. And how many percentage we are getting expressed in our genome? How many percentage of genome is getting expressed?
40.
Good. The 98% is generally. Pakka. They contain information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are called allele. I told you people are alternating form of the gene. Not always true. They are slightly different from of the same gene. If we use alphabetical symbol, I think you people know what they are talking about here. So you want to represent, you can take any symbol. The dominant you have to represent with a capital letter. Always the capital letter is going to come first of that particular gene, right? If you are taking other gene, you can write a small letter and that capital letter, doesn't matter. Got it? This one. So nothing is there to explain here. Now here. From the observation that the recessive parent trait is expressed without any blending in any F2 generation, we can infer that when tall and dwarf plant produce gamete, by the process of meiosis, the allele of the parental pair separate or segregate from each other and only one allele is transmitted to the gamete. That's what we have said, law of segregation. What we have said? During gamete formation, the pair of factors are this segregation of allele is a random process, and so there is a 50% chance blah 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 gamete contained. Yes, we know. In this way, gamete of the TT parent will have T only, pakka. A small TT will only have, and if they are going to fertilize, they will produce this one, right? And now this is going to produce two different type of gamete. Pakka. Since these hybrids contain allele which express contrasting trait, they are called heterozygous and homozygous. You people know. The production of gamete by parent, the formation of zygote, the F1 and F2 plant can be understood from the same thing, the same very thing in asked that question, yes or no? Exact the same sentence was there or not, where they have asked that which diagram can be studied with the help of which diagram, Punnett square, Bullet square. So few points are given there, yes or no? Same point is written. It was developed by a British scientist, Vignol C. Punnett. And to honor him, we are saying this is done by biology teacher. Right? It's a graphical representation to calculate the probability of all possible genotypes. Please remember. So Punnett square we are using to the guess the age of the person, right? No. Gender of the person. How will it? No, you people can eat. I don't know. See, in my graduation, I have a super teacher. he allows everyone to eat. You can take that rajma chawal, Maggi from the canteen, you can eat there. But don't disturb the class. You can do whatever you want, don't disturb the class and listen. For me, you can disturb the class and you answer. Now try and understand. Important here, the question may ask that with the help of Punnett score, what we can find? Written in NCERT. What we can find? Genotype. So we can guess the genotype with the help of Punnett square. The possible gametes are written on the two sides, usually the top, and you know how to write this, and male and female. Anything else important here? Yeah, these two, please remember. Biofilm class. The same thing explained here. No need. I think you asked this. No need of this one. Okay. This is nothing but the binomial expression of ax square plus by square. Okay, don't become engineers. Not that. Now, one thing I like to tell you people about test cross. We have not discussed two things. Test cross and back cross. We'll discuss today. Try and understand. What is test cross means? Test means what? You are checking something, testing something. Suppose you'll get a biology teacher and you don't know that he's a biology teacher or not. Then what you have to do? Ask questions, and that too basic questions. If he's answering, then good. Otherwise, no. The same idea applied here. Suppose we'll have in one case this type of individual, biology teacher. Now what you have to do, you have to always ask basic question. The basic means you can always multiply with unknown. This is unknown. And this is the individual, you know that this is the homozygous recessive. Always you have to take homozygous recessive pattern. And then you have to multiply. What you are going to get? Modo. क्या मिलेगा? What you will get? अरे नहीं, क्या मिलेगा वो तो पता. Capital T, small t, small t. No, capital T. Then small t. Right? We don't talk anything else. Just now understand it. Let's take this is an unknown variety and we are going to cross with. Always we have this is fixed. Yes or no? Now again we'll do. What we are going to get? Capital T, small t, capital T, capital T, small t, capital T, small t. Now I want an answer from you people. What is the difference in this cross and this cross?
Says में 50.
Here we are getting 50-50%. Yes or no? The ratio is 1:1. 50-50 means ratio is 1:1. What about here?
100%.
All are same. Now this test class is for same to know the genotype of that unknown individual. To know the genotype of unknown individual. If you are getting 1:1 ratio, if you are getting 1:1 ratio, it means this particular guy is can teach chemistry, physics, bio, anything. Heterozygous, right? If we are getting all with the same, it means what? They are homozygous in condition. Clear? This is clear. Or I'm not saying homozygous dominant or homozygous recessive. Homozygous or heterozygous. So if you are getting 1:1 ratio, it means they are heterozygous. If you are getting all same, it means they are homozygous. This is clear. And for what it is being used? To know the unknown genotype. And what we are going to know with the help of Punnett square? Genotype only. Don't say with the help of Punnett square we can know phenotype also. Not given in NCERT. Genotype. Hello. You are here only. Understanding? Pakka? Pakka na? Shall I give a question?
Yes.
Now one more is there. What is back cross? Back cross means what? Back cross. Same as test cross is little brother, right? Because as little brothers have more freedom at home, right, compared to elder ones. So he has more freedom. There in test cross we have a restriction. What is the restriction? That always we have to go with homozygous recessive, right? Here you have a choice. You can go with any parent. You have a choice to go with any parent. Please remember, any parent. So suppose you are getting an unknown. What? Capital T, small t. Right? You can go with any parent means what? Either capital T or small t. Right? Any parent. This condition or this condition. Whatever the result you are going to get. That's the conclusion. Now there is a two good thing, right, which you have to focus on. This is called like test cross is used for unknown genotype to know what is the genotype condition there, homozygous or heterozygous. This one I want to know. The back cross is used for what purpose? The back cross is used for what purpose? Right? I don't know. One is genotype, then one is phenotype. Done? Online batch, you people are there here? Online batch?
Yes, sir. Yes, sir.
Okay. So they have done this with the help of test cross, right? So if you see the dominant phenotype or genotype unknown, so they have done the results and they have interpreted that unknown flower is homozygous, right? Dominant, whatever it is. And here they have said that half of the flower are violet, while other so unknown flower is heterozygous in condition. Clear? Done? So please remember this. Now this is most important part. Based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called as principle or law of inheritance. The first law is law of dominance and second is law of segregation. Right? Now here in law of dominance, he talked about only genes. Talked about genes, right? Nothing else. Characters are controlled by discrete unit called factors. Genes only, no? Factors occur in pairs, and in a dissimilar pair of factor, one member of the one pair member will going to dominate, another is going to be recessive. Right? The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 generation. And the expression of both the in the F2, it also explains the proportion of 3:1 obtained at F2 generation. So what I told the people, any character which is getting expressed at F1 is now called as dominant character and explains the law of dominance. Clear? Now law of segregation. This law is based on, you are feeling cold. Now what you are writing? Back में हाथ तो खाते टाइम हम लोग डालते थे इसको. How many of you have done this? That before recess only lunch
So
is
first
over?
period we come to
So in class 11, 12, this was our routine. And then in class 12 we start following a ritual that every day one person has to bring chocolate. Chocolate means that 1 rupee. So this morning scenario is just in front of our school there is a small shop is there, not canteen, shop is there. So give them 50-60 rupees, the whole dabba. So Before recess only, the whole classroom is full of rappers. We got good blessings from our teachers. Okay, law of segregation. This law is based on fact that alleles do not show any blending and that both the characters are recovered as such in F2 generation through one of these is not seen at F1. Though the parents contain two alleles during gamete formation, the factor or allele of a pair segregate from each other such that a gamete receive only one of the two factors of course. Pata nahi kya. Law of segregation. During formation of gamete formation, the pair of factor is going to segregate from each other, right? Which is nothing but law of segregation done by mono-orbit cross. This is important. And here the most important thing is this, what I told you. So in case of incomplete dominance, neither is getting at both the extremes. We are going to be in between, right? Because only one gene is functional. Now, explanation of concept of dominance. What does dominance means? And trying to get the gist of the chapter from here. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait, means phenotype, right? In a diploid organism, there are two copies of each gene, which is called as alleles. Then now these two alleles need not always be identical as in heterozygous condition. One of them may be different due to some changes that it has undergone because of like mutation they are talking about or recombination, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic form. Let us assume that the normal allele produces the normal gene that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for production of. Now what happened? What they are saying. Suppose you'll have this condition, right? Two alleles are there. Now this one is functional and it's going to produce a protein, 3D protein. This is not functional and no protein is being expressed, right? Then what could be the reason for not functional of this, not getting expressed of this particular allele, right? What could be? It means the normal or less efficient, whatever, suppose either first is nothing is being produced, it's nonsense only, nonsense codon appeared and nothing is being produced. Second is what? Something is produced but not the same thing, altered. Third is what? No, nothing is produced and something is produced. Then what? A defective one or the half one is produced. So a normal or less efficient, a non-functional enzyme or no enzyme at all. A non-functional means something is produced but nothing is produced. These three criteria are there. In the first case, modified allele is equivalent to the unmodified. Okay, it will produce the same phenotype trait, the result in the transformation of substrate. Such equivalent allele pair are very common. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be depend on the functioning of unmodified, the one allele which is not affected. The unmodified or functioning allele which represent the original phenotype is the dominant allele and the modified allele is generally recessive. Hence in the above, the recessive trait is due to non-functional enzyme or because no enzyme is produced at all. Clear? Then codominance, I think you people know very well what codominance is.
Yes.
Both allele getting expressed in equal. Multiple allele, you people know. Inheritance of two genes, you people know. They have taken two genes at one place. Nothing to be worried from here. No. Chromosomal theory of inheritance. Who is done this hard work of chromosomal theory of inheritance? Oh, in my knowledge it's Sutton and Bovary.
और
So Mendel published his work on the inheritance of character in this, please remember this year, 1865, right? But for several regions it remained unrecognized till 1900. Okay, so rediscovery of Mendelian, I gave you people homework also. Firstly, communication was not easy in those days, and now his work could not be widely published. Now it's so easy that we are not taking seriously any information. Secondly, his concept of gene or factor as a stable and discrete unit that control the expression of trait and of the pair of allele which did not blend with each other was not accepted by his roommates. As an explanation for the apparent continuous variations seen in nature, thirdly, Mendel approach of using mathematics, blah 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 blah. In 1900, three scientists, now important, De Vries, Cornish and Vaughan, biology teacher, independently rediscover Mendelian results on the inheritance of character. Also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe the cell division. And once you are going to observe cell division, you are able to see chromosome movements. And you are able to justify that the genes are going to be present on chromosome. If chromosome is moving, genes are moving along with that. So whatever Mendel said is the characteristic of what? Genes, and they are justifying the chromosomal movement. Right? Okay, this led to the discovery of structure in the nucleus that appear to be double and divide just before each cell divide. This is called chromosome colored body, as they were visualized by Staley. By 1902, the chromosome movement during meiosis had been worked out. These two stupid guys, Walter Sutton and Bovary, noted that behavior of chromosome was parallel to the behavior of genes, and this is the one point you have to remember for chromosomal theory of inheritance. Clear? Then chromosome movement to explain Mendel's law. Recall that you have studied that behavior of chromosome during meiosis equivalent division and during meiosis reduction division. The important thing to remember are that chromosome as well as genes occur in pair. The two alleles of a gene pair are located on homologous site on homologous chromosome. This we know, right? One from mother, one from father. And this is G1, G2, all this. Right? Cell cycle event, nothing else. This people have find out yesterday only. Others. Those were the. What is A? What is B? Yes, online batch. What is A? What is B? Can you please help me? What is A? What is B? बताओ.
A chromosome.
And B is G, right? Online batch. पढ़ रहे कितना टाइम नीचे देख रहे हो अगर इन पेयर्स the difference is here, right? पक्का वहाँ से पढ़ लिया
अरे वहाँ पे भी तो लिखा है ऊपर chromosome and gene.
Okay. Online batch. Hey, people are here only. Devra.
Yes, sir.
Okay, dear. So, what is A? What is B? People are not replying. No. Find out. Again, homework. During anaphase of meiosis 1, the two chromosome pairs can align at metaphase, plate independently of each other. To understand this, compare the chromosome of 4 different colors in the left and right column, and along with that, read the cell chapter, cell division chapter. Right? from class 11, you'll get that. Yes. Sutton and Bovary argue that pairing and separation of pair of chromosome could lead to the segregation of pair of factor they carry. Sutton united the knowledge of chromosomal segregation with Mendelian principle and call it how during cell division chromosome is moving. Right? Following this synthesis of idea, experimental verification of chromosomal theory of inheritance by Morgan and his colleague led to the discovery a basis of variation that sexual reproducing during sexual reproduction produces. Morgan worked with the tiny biology teachers called this root flies, Drosophila.
एक बहुत गंदा क्वेश्चन है
बहुत गंदा है वो क्वेश्चन
कि जैसे फीमेल्स XX होते थे हम लोग एक इनएक्टिव होता है वो तो ठीक है अगर हम मतलब माइक्रोस्कोपिक लेवल पे जाके ऐसे XX बॉडी को मिला के प्रोड्यूस करना चाहें तो नहीं हो सकता मतलब कि
आप कह रहे हो कि दो क्रोमोसोम को मिला दो
कि XX होता है ना अगर दो फीमेल के एग्स दोनों XX हो तो हमको पता चल ही जाएगा क्या कि X है कि है ना पता चल जाता है ना
नहीं कहने गया था ये हो गया XX
हाँ तो जो ओवा होता है तो वो एक्स बॉडी कैरी करता है ना एक कैरी करता है तो अगर हम दो अलग अलग फीमेल्स के इससे अगर हम फीटस डेवलप करना चाहें फीमेल वाला तो वो कह रहे हैं कि दो एक्स को फ्यूज कर
you want two eggs to be there, two eggs from two eggs to be fertilized, right? Maybe possible, but how you are going to fertilize? Ultimately, you have to take that particular nucleus and then we can do that. No problem. But you have to take the egg of one female and nucleus of other female insert. Yeah, definitely. Which were found very suitable for such studies. They could be grown on simple synthetic medium, blah blah blah, linkage and recombination study. So Morgan carried out several hybrid crosses in Drosophila to study genes that were sex linked. Okay, so that's why he has chosen the autosomes, sex chromosomes, XY, XY, right? And the character he has chosen body color and eye color, wings and body color. The two crosses we have, right? So, the crosses were similar to the dihybrid crosses carried out by Mendel's in P. For example, Morgan. Dihybrid yellow body, white eye female to brown body, red eye male intercross their F1 and then in F1 we know that the progenies are exchanged, the male and female, here females are mutated one in F1, the males are mutated one, females are normal because of dose compensation, right? They have one X working. He observed that the two genes did not segregate independently of each other and they have two ratio deviated very significantly from 3:9:3:1 ratio. To explain this, he said that linkage and recombination are the reason for this deviation of ratio from 9:3, 3:1. Right? Linkage means what? When genes are present on chromosome. Recombination is what?
Cross.
If you have seen what is this? How many people have seen Shaktiman? Not in your generation, right? In our generation, on Sundays, we like when in like village there won't be any electricity, right? So we'll go to that people who have this white color TV and battery because that time inverters are not there, no? So color TV cannot be run on battery. So we'll go to that. To see two things. In morning Krishna is coming and afternoon Shaktiman. So there Samraj Kilvish is there. Kilvish, the bad person.
The
Villain. So he do like this, right? Not the number. It's crossing over.
वो शक्तिमान क्या है
And at last there is some good learning. छोटी छोटी मगर मोटी बातें. Remember?
सब मतलब शक्तिमान के लास्ट एपिसोड में क्या हुआ था यार
And we were tiny people then.
Tiny people.
Morgan and his group know that genes were done now. And one more person name is here. Please remember his name. Where it is? Just a show. Alford.
सर थोड़ा नीचे फोर्थ नीचे से फोर्थ लाइन में
Yeah. Student. His student is there, and he is able to develop that map. Okay. This year only came. What's the time? 21. Half an hour. Too much time. This is cross 1, this is cross 2. We are going to get the polygenes. No, before we can discuss polygene, we can talk. That's too much time. Yes, Alan Batch. What do people are doing? हाँ क्या इसके बाद बता दो हम ऐसी सूचना पहुंचाओ मेरे कान तक पहुंच जाए
मुझे खाना कब चाहिए तुम्हारे
ठीक है वो उन सबको तो खाना घर पे ही मिल जाएगा अभी खा भी रहे हैं मम्मी ला के दे यस डियर यू पीपल आर एंजॉयिंग एट होम नाउ अंजना भाविका गायत्री मनाल नॉट एंजॉयिंग
हम भी मना ही करते हैं
Serin, I heard that.
We're not enjoying.
Yeah, I know. I heard that sound of utensil. That means you are eating something. And see, here people are discussing what to eat.
No, my Dada wants to eat.
Oh, okay. No problem, dear. You can eat along with them, na? So, super cool. Then Gayatri.
Yes.
Not eating anything, not enjoying at home.
No, sir.
No, sir, by smiling. That means yes, sir. Good. Enjoy? No problem. Now coming to offline batch. Not enjoying. Enjoying? At home, I'm asking. What time you people are studying?
सर 9 to 12 कोचिंग से साथ में
रात में? Night?
हाँ
3 घंटा.
नहीं मतलब वो डिपेंड करता है कितना लंबा टॉपिक है आपने बोला ना 1 घंटे बैठे नहीं
7 to 10 then डिनर करके 11:30 से 2:30
With phone.
सर फोन तो मम्मा ले लेती है घर जाती
अरे
पढ़ना जब तक पढ़ना होता है तब खाना खाते टाइम दे देते हैं
Then lunch, dinner hour become bigger. Right? How much time it take for dinner?
हाँ फिर मम्मी कहती जल्दी खाना
How much time is study?
सर रात को मैं 12 बजे से पहले तो सो ही जाती हूँ उसके बाद नहीं जाती मतलब पहले जाती थी बट फिर वो पूरा हो जाता है फिर सुबह उठ के 6 या 6 बजे उससे पहले नहीं उठती तो इतने टाइम में जितना हमें टॉपिक जो भी कवर करना है सब इसी टाइम पे कर लेते हैं
सर 9 to 1 या 2
जब मन करता है तब ऐसा कुछ नहीं है पढ़ना क्या फोर्स करना है पढ़ने से कोई राजा बनाए ठीक है ये मतलब सही है जिंदगी की प्रक्रिया ऐसे ही सुपर यार हाँ
7 to 10.
क्या 7:30 to 8:30
7 to 10 और फिर मॉर्निंग
10 बजे के बाद सीरियल
डिनर वगैरह
बनाते हो खाते हो
खाता हूँ
इतना टाइम लगता है
साथ बनाते हो
तो लगता है मैं बना
तब तो
इसीलिए जो है परिवार के साथ रहना चाहिए खाना टाइम पे
काम हो जाता है
don't have to do anything just have to show that you are busy everything will be done and I master in that okay so
आप क्या कर रहे हो प्रिटेंड करते हो
फोन पे रख के बोल
yes,
दिया
business people have seen that name that I
This is
think
business.
no he said like business so please increase the online batch how you people are studying Bhavika Gayatri and Sirin How much time you people are studying? Now they will think.
कौन सा टाइम बोलूं? बेस्ट आंसर
Best time. Then
Sir, night owl.
night out?
Night
मतलब
hours.
रात में
Night out.
Yeah.
But timing dear. Timing.
So mostly at night, after the classes I study and I take break when my mommy calls me for taking lunch at 6. And after that I study from 7 to 10. Then after that dinner, again 1 hour, then again sleep.
Super dear. Good. Just increase the timing, right? Gayatri,
No sir, even morning slot is booked. I sleep near about 1 hour 30 or 2 and then get up early in the morning, like not so early but by 7 or 8, and then I study till 6.
good dear. Gayatri and Bhavika, you people are there?
सही से
Huh? Yes, good job. Gayatri started typing. Are say, speak. You people are going to become doctors, right? You know, we're still ragging in the medical college, right? No? Yes. And still in medical college, still because your seniors are going to help you with your practicals, right? So it's not engineering that you just have a computer and everything will be done, right? Yes, Siri, say.
Sir, is having a time is something like very productive thing, because I don't think I have any fixed time, but I have this thing that I have to do this and I do it anytime whenever I feel like. I don't have
That
any
is the
proper
best.
time.
That is the best thing. That's what I said yesterday. That don't fix the timing to study. Fix the topic to study, and you have to complete that in that particular day. Then that's the best way to study. And slowly increase the time. At least I want you people by the month end to go for like 10, 15 minutes.
अच्छा
Hours. 10, 15 is what? Months. Which one is appropriate? Minutes, second, months.
बचेगा ही नहीं क्या
सर अभी अगर हमारी 2 क्लासेस हो रही हैं तो जब हमारी रेगुलर 3 क्लासेस या जितनी भी होंगी तो टाइम क्या रहता है 10:30 से 4:30 10:30 से 4:30
उसमें उसके बाद 10 घंटे और कैसे निकलेंगे
Why not?
आप कैलकुलेट
Okay.
सर खुद देखो ना घर जाने में 1.5 घंटा लग जाता है पूरा फिर खाना वाना भी खाते हैं
That's what. It's taking 1.5 hours, right? You are coming by running or taking help of some metro, right? Utilize that time, na? Take out the NCERT and start reading. That's why I told you people that every day I see 50 people, 20 people studying NCERT at metro station. Utilize that time, little bit time. So that time you are not doing anything. You are just judging people. I'll also judge. How come he's standing there? How come
Like
his
songs
legs on his head?
cup.
Yes or no? Songs you can listen, no problem. Songs are the best one, right? No problem. But utilize that time. One day you're not reading, no problem. You're having some emotional issue, no problem. But let's do it. In a month, if you're not reading 4 days, nah, but more than that. हाँ, और चलो कर कितना शाम से
आप 10:30 से 4:30 वाली क्लास
हाँ चलो ठीक है
सर 4:30 थोड़ी ना होगी सर फिर 1.5 घंटे के आसपास
चलो लिख दिया शाम को 5 बजे से ठीक है 6 to 10 done how many hours
4
right 1 hour for dinner make it half an hour as you are going to approach to the examination right and study while eating right don't google then what
देन 10:30 से
at least I'm saying 11 o'clock till 1 o'clock how many hours 2 how many total
6
6 How much more to go?
6 hours.
6 hours more to go. Sleep, wake up at 6 o'clock. 6 o'clock. Not 5, not 7, 6 o'clock. How much more time you have before you are going to start for day? 10:30 na?
So हम 9:30
How much more? 9:30 you are going to get in the train. So half an hour, 1 hour at least for coming and going 2 hour plus. Those people are going to come far away from, right? Then at least 2 hours from here now, 6 to 8, right? So 2 hour plus here and 3 hour from going and coming. 5 and 6, 10. Yes or no? And slowly these batches are going to shorten. Yes or no? As the course is getting over, so utilize that time also. What I said, 10 to 15 hours of productive learning. Got it? What is it? Online batch. Nothing. Gayatri, after class, I take a break. Super dear. Best. Okay, so we'll study polygenic inheritance. What is polygenic inheritance? Mendel studied mainly describe those traits that have distinct alternate forms, such as color of the flower or either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have a tall or short people as two distinct alternate, but as a whole range of possible types. Such traits are generally controlled by 3 or more genes and are thus called polygenic inheritance. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Please remember this, whenever we are undergoing any expression of a gene, 2 factors are going to be important. One is the gene condition, they are dominant or recessive, and second is environment. A person with the same genetic makeup, suppose two genetically identical twins, or you can say the zygotic twins are there, and you are being separated from birth. One is in India, one is in US or UK, right? Do you think they will look alike? A lot in their body structure, their complete expression, because it's ultimately the environment which is going to influence the expression of that gene. Yes or no?
Yes, sir.
So that you have to take care. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, that is the effect of each allele is additive. Please remember, in question we have used this only, right? That each allele is giving an additive effect. In question 5 gram, 5 gram, 5 gram. We are adding up now. It means what? Each allele is having an additive effect. To understand this better, let us assume that there are 3 genes A, B, C control skin color in humans, with the dominant form A, B, C responsible for dark skin color and recessive trait A, B, C for the light skin color. The genotype, this one have a darkest skin color, and that the recessive will have lightest one, and then the between one is going to form a gradient, right? As expected, the genotype with 3 dominant allele and 3 recessive allele will have an intermediate skin color. In this manner, the number of each allele is going to produce an additive effect, nothing else. Right? We have done a question on this. Pleiotropy. We have so far seen that these 2 topics were not there in earlier NCERT. They were introduced to new. We have so far seen the effect of a gene on a single phenotype. I told you people, just opposite of polygene is pleiotropy. There many genes, one character. Here one gene, many characters. So there are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called pleiotropy gene. And one we have to study. What is that? Phenylketonuria. Right. And the enzyme is given. Phenylalanine hydroxylase. Right? Sex determination, you know. Huh? मैंने
तो वहीं से बंद कर हो जाएगा।
permanent delay. Here, three individuals are given with XO and XY, right? So XO means in insect case, XY means we are talking about some other individuals. So here we have XY, XY, and here we have female heterogamity, right? ZW and Z. Then sex determination, human, honeybee, I told you people, the fertilized egg is going to form female and fertilized egg is going to form male.
रॉयल जेली कहाँ से मिलते हैं इनको?
They have in the same something they form called as royal jelly, which has been fed to it. Like same, the person who is going to become a king, they drink some water and become royal water. Maybe the consequence may be different, I'm not saying, I don't have any knowledge in that, but same. Mutation we have done, genetic disorders we have done, Mendelian genetics we have done. This is important. Please don't forget. Hemophilia, sickle cell anemia, how they got this shape, phenylketonuria, thalassemia, and chromosomal disabilities, aneuploidy or euploidy or polyploidy. There we have three: Down syndrome, Klinefelter, and Turner syndrome. With this chapter is done. Any doubt, please let me know. No doubt. You can do any question from this chapter.
Chapter sandwich खाना?
हाँ? क्या?
Sandwich.
Sandwich. अभी नहीं, बाद में यहाँ से लेके बात करूँगा. Thank you dear. No, you can enjoy.
अरे नहीं मेरे पास एक्स्ट्रा है इसलिए.
You have asked, that's too good for you. चलो, अब क्या करें?
सर
छोड़ बोर नहीं कर सकते 1.5 घंटे तक.
सर आपने बोला था ना कि आपको और अच्छा इंसान बनना है, और आपको क्या करना है सर? आपको और
No, see, whenever you touch the feet of elders, what they say, become a good person. But then I realize what does that mean. It means become a good person. What else? Any question? Or online people, you understood Hindi? Bhavika, Gayatri, Srini.
आपने गलत आंसर दिया. मैंने पूछा था आपको और अच्छा मतलब क्या करना है, like in general.
करना है नहीं, मुझे अपने आप में और अच्छा बनना है. मतलब अभी जो कुछ मेरे अंदर गलत बातें आ जाती हैं वो ना है, वर्क अपॉन वो तो नहीं रिमूव हो सकती. नहीं तो आदमी भगवान हो जाएगा, लेकिन फिर भी कुछ ना कुछ तो रहेगा, तो उसी पे काम करना है. ऐसे और कुछ
So for life में when was a point where you were the most angry?
मोस्ट है.
मतलब most happy तो सभी पूछते हैं.
कुछ इंस्टेंसेस ऐसे होंगे, मतलब for 5 minutes maybe and then went away.
सामने स्कूल के बच्चे को
नहीं, never. कभी भी क्लासरूम में किसी बच्चे कभी ऐसा मौका ही नहीं है.
सर अब आप आगे की क्लासेस क्यों नहीं लोगे?
मेरा मन नहीं कर रहा था इसलिए.
तो फिर अपना जॉब
काम तो करना
करना पड़ता है इसलिए.
नहीं, ये रीजन है, कुछ था उस पर डिस्कस नहीं कर सकते.
वही तो जानना था.
कोई नहीं, मैं हूँ यहीं पे, मैं आप लोगों के साथ रहूँगा. मैं कुछ कह रहूँगा, हेल्प यू पीपल. कभी भी आओ. And then we are going for YouTube also. मैं ट्राई करूँगा कि क्लास 12 के लिए टॉपिक. How you feel about anatomy chapter? Good or bad? Flowering plants.
सबसे बेकार चीज है.
Why? All and bad.
जेनेटिक्स उससे बेटर है.
What do you
उससे बेटर है. सब कुछ उससे बेटर है. वही सब कुछ उससे
There are not many of flowering plants. So super good.
सर स्टार्टिंग का तो ठीक है, इतना कुछ खास नहीं है. जो लास्ट का पार्ट है ना, सेकेंडरी फ्लोएम, सेकेंडरी जाइलम, ये वो
इजी है स्टार्टिंग का.
हाँ क्या मैं ही उल्टी हूँ क्या?
Okay, we'll start discussing. Let me know what could be the definition of a tissue.
Similar type of
Similar
cells
cells
performing
performing
a
a
function.
performing a function.
It's called tissue, right?
हाँ.
कुछ और इसमें anyone wants to add something to this, that similar type of function being done by few cells, right? Tissue? I'll give an example. We say that cells are building block, right? It means suppose we have to construct a wall
Similar cells with intercellular space.
with intercellular spaces. Okay, correct here. But that intercellular space is what? Suppose cell tissue is going to be consist of cell, right? So it's going to be consist of cell. It means what? If suppose I'll give 200 cells to you people, right? You have 200 cells, right? Do you think it's a tissue or a 200 cells collection?
200 cells, 200 cell collection.
Right? Second example, let's suppose cells are equivalent to bricks and we want to make a tissue that's equivalent to wall. So just you're going to put bricks one above the other, that's a wall or what? Do you consider that's a wall? What you required there?
That's not.
Cement.
Cement.
Again,
There
we
must
require
be
cement
some
exact.
cementing material which is going to hold the cell and that's called as ECM. What is that? Extracellular matrix, where the cells of tissues are going to be made. Getting idea? So a tissue definition is going to be a group of cells which perform similar function, may have same shape or origin, which are put together with the help of extracellular
Matrix.
matrix. Okay? Understood? Now if discussing about plants, how many type of tissue plants are? Meristem and permanent, right? So one is and one is permanent. Meristem means they have the division capacity. Permanent means they are differentiated cells. They don't have the capability to divide. Okay? Now try and understand. Have people seen that Three Idiot movie where he challenges the virus itself? The Dean to find out two word meaning. Same? Can you see something here in this meristem means what? Same thing, same phenomena here. How many people know about stem cells?
Yes, sir.
What is stem cells?
जो they can convert to
Who are themselves undifferentiated, they divide to form many different cells. Yes or no? So plant body is developed from stem cell. They have the characteristic of stem cells.
सारे organisms होते हैं.
सारे होते हैं. In plant, in animal we say stem cell. In plant there is something, so we add many stem cells. Got it? And from stem cells, permanent cell is going to form. In permanent cell there are two types. One which is consist of same type of cells. Parenchyma, collenchyma, sclerenchyma. Or complex consists of many different type of cells. Xylem and phloem. Right? Parenchyma, collenchyma, sclerenchyma. Parenchyma is what? Village. Collenchyma is town. Sclerenchyma is metro city. Means what? Parenchyma, village life. People are living, they have a healthy relationship. Yes or no? They care about each other. Right? Like we care in metro. Right? Seeing an old person, offer the seat please. And hope that now he is blessing. He didn't do anything. He's taking out phone and start playing game in front of me. I'm not playing, that guy is playing. Right? And they start getting shit. This person is younger than me only. Right? So in village people are living. there is a gap between houses. Yes. So does the parenchyma cell. Cells are living and there is space between cells. और अरे क्या? कोई बात नहीं. चोट तो नहीं लगी? Now understand. Then comes the collenchyma, town. Collenchyma means what? Town means what? From village to the town. The spaces are not there because land cost is more. Right? Then, but still they are living. They take care about their neighbors. Yes or no? And now comes the. That's why collenchyma is living mechanical tissue, petiole, green color. Right? And then comes the sclerenchyma, metrocity. Nobody caring about anyone. Chichi. Dead. Dead. No space. Nothing. Yeah. So this is the characteristic. Then comes the. What is the super chapter? Read NCERT. Then. Which chapter? Biological classification. Panjai.
सर आपने बायोलॉजी क्यों चूज किया
I don't know. Maybe because in class 9, 10, my teacher was super. So I don't have to open the book itself. Whatever is said. So I developed this too much interest for the biology. And in class 11, 12, I got a teacher. Whatever he said, I didn't get. And from morning, the time the sir is going to enter in the class and the time he is going to get away, continuously he is going to teach only. And not even a single word is going inside my brain. And that is the time, right, we become like mature and start seeing world, right, because there is a freedom from home and lot many things. So nobody caring also. Biology only now we can do. And then we know we can't do. In BSc I starting buying myself. So it's because of that teacher. Rajesh.
मेरे पापा का नाम भी राजेश
No, he was a super brilliant teacher. He didn't write anything on board, just front of class, and this finger is like this. So I know 1, 2, and this is like 3. That finger is always wrong. Super awesome.
सर आपने भी PhD की है?
No, sure. Why to do PhD? They are in research, 30 year age. Done. Then 1 year I have done. Thank you, sir. Thank you for this beautiful opportunity.